So welcome to video 3 in our course on phylogenomics. So here we're talking about sequence alignment and optimality criteria. So if we want to think about constructing a tree like this, we have to consider how should we construct a tree related to the optimality criteria being used, and also how are we aligning that tree in the first place to create a sequence alignment that's informative for our particular um, set of data that's of interest. So if we go back to alignment, we have a particular set of input sequences, and we'll consider what dynamic programming algorithm are we using to compare these different letters against each other. So this example is for amino acid sequence, but the same applies if we're looking at DNA or RNA sequence as well. So we can have some kind of more easy examples like this one, where it's kind of easy to infer what the patterns should be. But then when we look at other examples where we have our alignment of fat cat sat on the mat or fat cat sat on the cat, what we can see is that if we compare, say, tea coffee versus Cristal, they have a slightly different approach for these two alignments, where we have kind of trivial set of three sequences. But we see here that with this AT sequence here that can be placed at either region, that tea, coffee, and Cristal have a different approach to resolving that question. And in reality, we don't know which is most correct. It's just different approaches. They have really the same alignment scores, whatever way you want to look at it. But you can see that it's different, a different approach for each particular one. And therefore, for any particular alignment, once it's big enough, we can identify many different solutions for that. If we look at this other example related to Garfield, the last fast and very fast cat, you can see that we have different sequences represented here, and therefore we can look at the patterns that are of similarity across the different sequences and see which among these two are most related or which are more divergent. So we can kind of see at the start that we have differences here at these four letters, where we just have one change here at L and F for these sequences A and B, where this is four changes. We got some similarity here across F and C, F and A here, then an S insertion, and then an indel here. So it appears that A and B are more related to each other, but C is not too divergent from A, but C is more distant from B. So what we can do is denote this in Newick format. So we might say A comma B with round brackets around it, represent the topology, and then add C onto it like this. And we know that D is more divergent, so if we can add D as an A group like this, just putting a comma between these different ones. So that denotes Newick file formats, and that's a readable format. And we can add information in here on distances into this particular arrangement, which we'll look at later. And therefore, we can reconstruct at least the topology of these different samples in this manner. But note, this is a cladogram. It does not represent the actual information contained in the terms of the numbers of mutations that occur with these different samples. So we need to do apply inferential methods to actually deduce those quantitative distances. So in principle, regions that look at orthologous regions with different samples can be alignment or graph based. So you're probably familiar with alignment tools. They align sequences to produce a tree that might tell us about marked regions that have a peculiar origin. This can be difficult if the ancestry is very ancient, if there's horizontal gene transfer, if there's incomplete lineage sorting, that can be harder. So graph-based methods can be better when we're looking at very, very divergent samples. So this is based on relative similarity. So maybe doing reciprocal breast hits between different regions of a genome and trying to identify ones that are one-to-one orthologs between different segments, and then taking those segments that have that high matching rate, and then aligning them to produce a kind of an average tree for that particular set of specimens. So this might only use part of the genome in our assessment, whereas if we're dealing with alignment-based approaches that are working with more recent samples, that might take a larger proportion of the genome. So alignment-based approaches are also typically Bayesian and Bayesian evolution-based approaches are typically progressive. So they start with the most stable set of samples and then gradually add a sample into that to broaden out the alignment both in terms of the number of samples and the regions that are aligned. So MAFT, T-Coffee, ProgCons will be examples of those tools. And they do approximate alignments to create a guide tree that then feeds into the next alignment stages gradually. We also have ones that look at the consistency of the alignment. So MAFT, muscle, and Clustal are example of methods that apply this. And to do this, they do the more exhaustive approach of doing all possible pairs alignments, which is computationally time-consuming, and that allows them to retain high-scoring pairs between the pairwise aligned sequences. So there's other examples here in this table where you have common tools would be Clustal, Clustal Omega, Muscle, ProgCons, MAFT, that allow different methods to be applied. Which one is best is a question for your particular research question at hand. So if we're looking at phylogenies, we know that phylogenies for a single region are generally okay. That makes sense to do. But if we're looking at multiple regions, that might be complex depending on how much heterogeneity is present and how different they are. We can amalgamate these different regions together into what's called a supertree. So that's a set of phylogenies. Or indeed, we can perform the more common supermatrix approach, which is to amalgamate different regions together as input for the tree. So that brings us to optimality criteria and considering what different methods are best. So you have things like distance-based method, parsimony, maximum parsimony, and maximum likelihood-based methods as well. So consider whether the minimum evolution is the best approach. So that's the shortest tree based on the sum of all the different branch lengths. Maximum parsimony tries to minimize the number of substitutions on external branches and pushes them onto internal branches. Of course, when you have, for example, viruses that have multiple substitutions, independent substitutions at a particular site, that may be incorrect. And then there's maximum likelihood that try to minimize the number of different events that occur in a kind of iterative Bayesian approach, modeling the likelihood of the tree given the data. And then we also think about the search algorithm that we're looking at. For example, neighbor joining algorithm is most common because it may not be possible to test every possible tree. So that's also reality. So we think about then our optimality criteria, which is like our recipe, our code we write to determine the criteria we want. And then we have the appliances that we're using to search algorithms. And if we have more, say, compute power, we can use better search algorithms to search through more trees. But that's still dependent on the recipe that we're using. So we consider it in this way. We have our input data, our aligned sequences, which might be, for example, virus capsid regions. We have our search algorithms, our appliances like neighbor joining algorithm, and its approach is dictated on the optimality criteria built into our models, and that will give us our output or results at the end, our phylogenetic tree. So if we use a different recipe or a different appliance, that will give us a different tree at the end. So it's worth bearing that in mind. So we have distance-based or character-based methods. These essentially calculate the distances between each sample pair as a simple number. That creates a distance metric of the pairwise number of differences between different samples based on the numbers or on a fraction. Typically use neighbor joining to compute the best tree, but this is not effective if the samples are very, very distantly related to each other. Other methods are character-based. So maximum parsimony is the most common example of that, where we also have maximum likelihood and Bayesian methods that also apply this as well. Maximum parsimony tries to minimize the number of substitutions that occur on a particular tree by pushing more of them onto the internal branches. Of course, there's a limitation on that we have to search through different possible trees. So if we've just 10 samples alone, we may have, in theory, 8 by 10 to the 21 different possible rooted trees. And the issue with maximum parsimony is that it doesn't consider rate variation between different site classes. So transitions versus transversions, third coding sites might have different patterns of mutations compared to other sites. So what we're thinking about with maximum parsimony is that we have we've got a single ancestor in our tree, and each substitution has one origin only occurs once in a tree, which might be true for certain organisms but violated for others. It pushes the mutations onto internal branches. We want to retain the sites that are parsimony informative only. So ones that occur in two different samples are informative. Ones that occur in a single sample only are not informative because it only represents information on that single sample, and therefore does not tell us about the collection of samples that are at hand in a general sense. So what it seeks to it optimize on the basis of minimizing the number of mutations that are required to explain the particular tree. And it's effective with closely related sequences. More distantly related ones, it may be less effective. So what we might do is create a quick tree at the start. Sometimes you can have a predefined user tree as input to guide that. It commutes the different branches and computes the parsimony score based on that. So it tries different trees and sees how many substitutions are present across this different tree, and then reconfigures this and ignores ones where they have extra mutations that are present here, or tries to minimize that number of mutations. So this is an iterative approach that explores the three different tree options that are possible. So you've got a representation of this here, where we got three possible tree topologies for four outgroups that are present. So if we look at this particular alignment here of protein sequences, at site 1, we've got 1 and 4 are closely related, and 2 and 3 are closely related. And that's represented across these three different trees, where we've got 1 and 4 closely related here. It's like it fits site 1, whereas this is less consistent here because you'd have to have mutations occurring on this branch here and this branch here, whereas you just have one for this branch here, and the same for the other symmetrical option as compared to tree 2 here. If you look at site 4, where's site 4? We have 1 and 3 are closely related, 2 and 4 closely related. So therefore, this tree here on the right requires two substitutions, where this one in the middle with SS at either end and TT at either end requires one mutation, and this one on the right or on our left requires two different mutations. So therefore, the most optimal tree here is going to be this one here, based on the number of mutations that occur across these different lineages. And that's because we've got an invariant site here, and this site 3 here, 1 and 4 are more similar, 2 and 3 are more similar. So that's why you have 2 by 2 for that score and 2 by 1 for this score here. So we score the trees and determine that this is the most optimal tree. Another character-based method is maximum likelihood. So it looks at a particular model that's of interest, that considers how much branch lengths should vary, and looks at what's the chances of getting this particular data set given the model that's of interest. So maximize the likelihood of that. Examples of methods that use that are, we'll use MaxML later. IQ3, which is available in R, also does that. You may also, FastTree is also a good way of doing that if you have a very large phylogeny for which you want to estimate a tree quickly. And then PAMO is an older method for doing that. So depending on the context, maximum likelihood can be the best choice, but it can be computationally take some time to execute. And then we can also tweak it to have different algorithms similar to those used to find the maximum likelihood, maximum parsimony tree as well. And we have to bear in mind then for distance-based methods is that they don't necessarily take all the information that's available from the different sites, and certainly are inferior compared to maximum likelihood methods. There's also Bayesian approaches to solving this, and character-based as well. That needs a model and a likelihood function and a prior distribution to estimate that. It's typically based on Markov chain Monte Carlo methods to estimate the uncertainty. It considers the tree topology and parameters, produces a maximum posterior tree with credibility intervals. So example of this would be Beast and Beast 2. Mr. Bayes, and so on that take that approach. So that's another effective approach. So we talked about how we approach these different algorithms. So example of this is minimum evolution, where we minimize the sum of the branch lengths that are present. So we create all possible trees that are present, sum up their branch lengths, and take the one with the smallest overall branch lengths. This is tricky because if we have just 20 samples, we'd have many, many trees. So this is tricky. A more effective way of doing this is to use a neighbor joining algorithm, and this essentially is bottom-up clustering that helps to minimize the sum of the branch lengths that are present. And we'll look at this example of the UPGMA tree construction approach. So you know, for example, from our earlier discussion of the green samples, that they're more similar to each other. And therefore, when we're studying our phylogeny construction, we can make them more similar and then add the green blob one here as the next one similar to them. And we can see that we count the number of substitutions that are present, that the purple and pink samples are very closely related to each other. So we cluster them next before joining the green ones together. And then we can assess, are the green ones as a shared node more similar to the purple ones here or to the red ones? How does that join? Does that join here or does that join out here? And therefore we can count the number of mutations that identify that the red one is more similar to these two here based on the TAC, which differs above as well, and construct this sort of tree. So you've essentially got two different groups with different levels of similarity. So what the neighbor joining algorithm is trying to do with these samples is start with a star-like phylogeny at the start, where each branch has a maximum number of external samples, and take the two that are most closely related and merge their mutations onto this single branch, and then repeat that. So we might take another sample that are closely related, like 7 and 8 here, and create a internal branch associated with them based on the mutations that they share. So we can sort of see that there and gradually grow the tree like that. An example of this is given, this method was first developed by Sato and Nye in 1987, but it's been since been modified at least more than once, and there's different variations of the NJ algorithm as well. So what we consider is a tree like this, where we quantify the number of changes that occur across each branch with numbers. That may not be possible to do for all different tree types, but it works for this illustration here. And what we're doing here is considering which samples are most related to each other. So you can see here that A and B are have the same ancestor going back in time. And we can see for A and B that they have 5 changes between them. So what the bottom-up algorithm, NJ algorithm, does, it looks at the number of mutations that occur. So although A and B are in the same ancestor, when we look back in our tree, we see that C is more related to A than to B. So C has 7 changes compared to B, whereas only 4 compared to A. So therefore, this will be the first sample that is clustered together, because D and E have 5. F is quite divergent to the other samples as well. So start with that one, A and B together. So an example is given here, where we have ATG, GGA, there's a change there for A and B, another change here for A and B, 2 sites that change here, and therefore another one here, and that gives us 5 differences. We've got 1 change here, 1 change here, 1 change here, and therefore what we can see is that and change here that they have 4 differences between A and C, and therefore we can cluster those samples together first and merge those into a single node here and compute that distance between that node there to those for B. So this is known as the UPGMA tree, one-weighted per group method with arithmetic mean. Determines the number of pairwise differences between different samples, like those shown here, for example. So we can see here we've got different mutations that occur and different patterns of similarity between the different samples. So between sample 1 and 2, for example, we have C to T and G to C. So therefore, we can compute the number of pairwise differences between the different samples. And we see therefore samples 2 and 3, we only got one difference here at site 4. So what we can do then is do that bottom-up clustering of samples 2 and 3 together. So we got V1 versus V2. We can make a new node U from samples 2 and 3 and compare that distance of U to the other ones. So we're writing here is D for U compared to V1. And we compute that by comparing the distance between V1 versus V2 and V1 versus V3 that make up U minus the distance between the samples within that node. And we divide that all by 2 because we were looking at 2 different lineages when we think about it. So we're looking at 2 different lineages and comparing really to this U ancestral node against this sample here, V3. Sorry, V1. That's V2. And that's V3. And we're looking across this part of the tree here. And therefore, that's V1 versus V2 is 2. V1, V3 is 3. So we got 2 and 3 there. V2, V3 is 1. And that's how we get our value of 2 at the end. We did the same for 4 and 5. We get distances of 1 and 3. And therefore, the next step would be to cluster U with V4. So that would suggest V1 is not more closely related, that V4 here is more closely related. And therefore, we can construct an ancestral node here to which we compare V1 and V5. So that's the next step. We have this ancestor here. Cluster those together and repeat our clustering here. We're calling that node X in this instance. So we compare X to a V1. So you got V1 minus U, which is 2, V1 minus 4, which is 3, minus U versus V4, which is 1. So we get a value of 2. And V5 is more divergent. So you get a value of 3.5. And therefore, we cluster V4 with U, and we're comparing then the next stage is just that new cluster X versus V1. So call that Z, and we compute the distances in the same way. V5 versus X is where's V5 versus X? 3.5. V5 versus 1 is 2. And so we get these values here to give us the final branch distance length. So we can represent the tree in this manner, that we can see that V2 and V3 are more related, which we can represent here. And then we have V4, V1, and V5, which are represented in this tree based on the distances in that matrix. So you can see this representation of relative distances between different samples across this particular tree. What we consider then is slides where we have heterogeneity across a particular tree. So if we think about this example here, we've got a bundle of different samples here where there's a snip here at this G to A change here where B and C are more similar, A and D are more similar. Another site here, another site here, another site here, another one here, here, and here. And therefore we can construct our distance matrix in the same way as before, where A and D have the fewest number of changes between them, just at this site here only. And therefore they're most similar to each other. And therefore, when we look at A and B and C and D, when they're constructing a tree, we're essentially placing these mutations on these different trees. So we got a distance between A and D here, and then A and B and A and C and D. So we can kind of use different methods to look across the alignment, but bear in mind that we might have heterogeneity, that if we split these up, these portions might get different answers if we split them here, because we see that A and D are identical and B and C are very similar, whereas down here we see more divergence between C and D and B and C. So please look at the next video on bootstrapping that extends this testing of how robust an alignment is.